Good afternoon. I would like to thank the organizer to give me the opportunity to present here some results of this project that is focused on, just a moment, cell cytoskeleton as a target for ionizing radiation dosimetry. This research is supported by Museo Storico della Fisica e Centro Studi e Ricerca Enrico Fermi, and is in collaboration with the Trento Institute for Fundamental Physics and Application. As I have already said, the project began in the 2017, so it's quite new, and the investigation of this is about the investigation of cell cytoskeleton as biological target for ionizing radiation. In particular, we would like to correlate the different relative biological effectiveness of X-ray and protons with a quantitative parameters for ionizing radiation. The relative biological effectiveness is the ratio between doses of two different kind of radiation and connected with the effect, the biological effects. So, okay, no, okay, yeah. What is the point of this research? First of all, in the biological question is that in clinical use, when protons are used for radiotherapy, the RBE is more or less 1.1. And this is independent from the volume that is irradiated, is independent of the dose rate, and therefore is necessary to understand much better what is the different biological effect of this irradiation on the cells. In particular, we are focused on normal tissues. Normal tissues because the normal tissues, the normal cells, the not tumoral cells, are the cells that are surrounding the tumors, and they are exposed to very high doses. Also, these are not tumoral cells. In particular, this is the case of breast cancer, where usually the tumors is surgical removed, and then some radiotherapy is done in the same region of where there was the tumor mass, and actually is done on normal and not tumoral cells. So it's quite important to answer some open questions, understand what is the effect of this radiation on biological tissues. For example, there is this bystander effect, or the invasiveness of the cells, or at least the occurrence of a second tumor in the region nearby the first one, so recidive. For this reason, we decided to study to try to study a quantitative parameter that give information about the cell damage. The most studies structure is actually DNA or nucleus, but this is not the only one important structure for the cell life. Actually, there is also cytoskeleton, and in particular, we have some structure that are microtubules and microfilaments. The microtubules have a diameter of 25 nanometers, and they are involved in cell movement, cell shape, track for organelle movements, spindle for mitosis and meiosis, and as you can see are green here, and they are starting from centromere, and they run towards the external part of the cells. While microfilaments are mainly done by actin protein, they have a diameter around between 4 and 7 nanometer. They are also involved in cell movement, cell shape, organelle movement, and muscle cell contraction. So now I'm going to details 4 steps that were performed to carry on this project, this experiment. So the first is about protocol setting of proton and X-ray radiation. The second regards the sample preparation, so cell seeded, irradiation, membrane denaturation, and cell fixation. And then I will show you the atomic form microscopy measurement. And I will give you some hint about computational analysis of AFM images to identify these dosimetric parameters that we would like to use. So concerning protocol setting, this is a scheme. There is a proton beam. This is a Petri dish. And we have a cover glass that is covering the bottom of the Petri dish. The proton beam has an energy of 150 MeV. The dose raise is 1.2 gray per minute, and the beam side is about 6 times 6 centimeters squared. This is the experimental room. This is a proton gun. So, and here we have the Petri dish. This is another image. And you can actually see this laser to align all the setting. So, what about sample preparation? We are using these cells that are HS578 BST cells. These are human, mammary, not immortalized, and not a tumoral cell. So is what we have nearest to normal tissues that we can have in the lab. So in fact, it's not so simple to keep them for a long time. By the way, we seed them, and after 24 hours, we irradiate, and then we denaturate the membrane. It has to point it that the membrane denaturation is a mild denaturation. This is to preserve actually as much as possible the cytoskeleton, and to avoid protein denaturation of the cytoskeleton itself. And then we have done cell fixation in a chair in a biological cape for 30 minutes. And now I will show you some image of AFM measurement. We have just a moment, Park XA100 in our institute, and with this microscope and with this preparation, we could have quite nice images of this cytoskeleton structure. Here in the middle is not very well visible, but is an optical image taken with the optical microscopy, and actually is stained with Giemsa to see a bit better where the region has been taken. And okay, here we have the representation of 3D representation. So what happened to control? So these are the picture of control cytoskeleton. And as you can see, there is a very defined pattern, quite complex, but regular, I would say, and with different direction of these fibers. So some of them are just zoom in, and this is a kind of pattern that we would like to try to quantify with a parameter. And what happens if we irradiate with 8 gray protons? It's clear that something happened. They are very different images. We lose completely the order, the pattern. We see some brightest point that actually may be some aggregation of protein. And these are other images of these cells. But what happens if we irradiate with X-ray? So the setting is very similar. We have an X-ray beam. We have a petri dish. On the bottom of the petri dish, there is a cover glass with grown cells. And the dose are 25 gray, and the voltage is 195 kilovolt peak. And the preparation of the sample is the same. And okay, here again, the control. And this is actually the X-ray. Completely different pattern, completely different response to the cells. So the first impression is that there is more order structure, even respect to the control. This more ordered structure seems due to the lack of smallest fibers, and in any case, this rearrangement is completely different for respect to protons. Okay, now I want to go to some hint about computational analysis of these images. Where we started, so at the beginning, we decided to study a parameter called, that we call modified directional index, that is simply done using the Fourier transform, and is a parameter that is close to 0 when we have aligned fibers, and is close to 1 when there is disorder. This parameter is actually very good when all the fibers are aligned in one direction or in few directions, but is not responsive if we have a disorder number of direction or fibers. In any case, we at the beginning try these parameters also on not denaturated cells. And we have this control that is not denaturated. And then we have some X-ray radiation of non-denaturated cells, X-ray for gray. And why we want to use this parameter on a cell that is not denaturated? Actually, from the images, we could see that we could reveal, in any case, the cytoskeleton. It's like to have some tendon in your hand, and you have a skin on it, and so you can see the tendon, also there is a skin. So it's like the envelope or the convolution of, let's say, skin over tendon. And the same was in this case with the cytoskeleton. And in fact, using that parameters and choosing a region of the cells where the cytoskeleton has the same direction, was clear that there was a difference between this parameter number of the radiation respect to the radiation respect to the control. So the control was around 0.5, while the radiation was around 0.8. I just remember to you that nearest 0 is more alignment. What about if we denaturate? As I told you before, if we denaturate, this parameter doesn't answer anymore. And in this case, we see that the fibers are aligned with the same direction in both cases. And what is nice to notice is that also when we treat the denaturated cells, the parameter is around 0.5, so the same value more or less of the control. So what this parameter said? This parameter said actually that the cells were damaged, the membrane were damaged, probably with collapsing of some structure. And now we just, okay, just 1 minute. I have. We are going to another approach using the half transform. The half transform method is a method to identify region with defined geometrical shape. and is possible to have a layout determination, recognize trade segment, and it's possible to the assessment of fibers, densities, and connectivity. So what we are doing now is using this approach and through half-transform, identify structure, collect them, and set suitable parameters to recognize this even smaller structure. And so, conclusion, these preliminary results depict the cell cytoskeleton as potential target for ionizing radiation, such as DNA. X-ray seems not to cause the same damage magnitude that protons. Half-transform is a promising method to quantify the cytoskeleton damage and perspective. Classification of cytoskeleton damage into irradiated cells to possibly use of half-transform. Exploration of plasmatic membrane, but also investigation of cytoskeletal damage of tumor cells as possible involving into cancer invasiveness, and also some measurement with actually confocal microscopy to merge the data of AFM. Thank you.
Thank you for the nice presentation. Will there be any question?
Okay, so thank you very much for this presentation and interesting results. Could you comment on how you prepare your sample prior to the AFM investigations? Is it in dry? Is there any, let's say, observations that you think that could be relevant for the preparation, or?
You mean because we're fixed at the beginning?
I'm not a biologist. I really have no idea on how you prepare the cells.
Me too. Okay. At the beginning, we cover with paraformaldehyde, and so we had some result with this that he usually used to study the kind of things, but we had much better results with drying in air, with just a flow of the air. Actually, was incidental, these things, but this was the better resolution we got, probably because when you do AFM measurement on paraformaldehyde, you don't measure directly the fibers, but you measure the interaction with this compose. So I think this is okay.
Any other question? If not, thank you very much, and we can go